Alright, this is our first video on dihybrid crosses. I'm imagining it might take more than one simply because I really want to hash out each little step in my explanation of this topic because it is a tricky one. So we are inching closer and closer to the end of this standard. We've basically got this little topic, dihybrid crosses and linked genes, and then our kind of evolutionary subtopic, the population change connection to genetics, and that's it. So today we are going to hopefully differentiate between monohybrid and dihybrid crosses, describe how unlinked Mendelian genes can be segregated into gametes, and explain how fertilization of these gametes leads to a 9:3 to 3:1 phenotypic ratio in dihybrid crosses when we're looking at complete dominance. Now hopefully that sounds like a load of rubbish to you, otherwise it wouldn't be a learning intention. So this is what we are going to learn today. So just to recap what we know about the word monohybrid, well, the prefix mono means one. So when we've discussed monohybrid crosses in the past, it's been because we've been looking at the inheritance pattern of one gene. So even all of our non-Mendelian inheritance patterns that we learned about, we were always just dealing with one gene at a time, whether it was fur color or fur length or eye color or blood type. All of these were singular case studies, even if they had lots of different alleles doing different things. Okay, so everything we've done up until this point has been monohybrid, whether it was following the Mendelian rules or not. So your definition here is that monohybrid cross is the possible combinations of alleles for one gene found on one pair of homologous chromosomes. So when we use the word cross, we're talking about breeding two individuals with each other and seeing what occurs in the genotype and phenotype of the offspring. So again, we've just been looking at singular genes at a time, and we should know by now that, or we can at least figure it out if we're asked to, breeding two heterozygotes, so two individuals that have two different alleles, will lead to an expected ratio of 1 to 2:1 in the genotype. Okay, you can see that in the Punnett square there. If we have two individuals that are heterozygous, I'm not sure what the gene is here off the top of my head, so they're both big B, little b, all of the babies are seen in the middle, and we've got one homozygous dominant, big B, two heterozygotes in the offspring, and one homozygous recessive. Okay, so we can see that it's petal colour that we're looking at. Right, and that's the genotypic ratio. Obviously, the phenotypic ratio depends on the interaction of those alleles, whether it's complete dominance, incomplete dominance, co-dominance, blah, blah, blah. Okay, so in this case, it looks like we've got a complete dominance pattern where the flower will only be white if it receives two of the recessive alleles. Okay, so that's a monohybrid Punnett square, and we always get a 1 to 2:1 genotypic ratio, an expected ratio in offspring. It doesn't always hold true. All right. Oh, I see that I've added a picture there for the gene that we were talking about. I just didn't look at it in time. It's been a year since I covered this last. Okay, lots of information on this slide, but I will go through it bit by bit. So obviously we just discussed the word monohybrid. Hopefully we can deduce, using our powers of inference, what the word dihybrid means. Di usually refers to two, like carbon dioxide, for example. So simply a dihybrid cross, or breeding of two individuals, will show us the possible combinations of alleles for two different genes. So we're looking at the offspring of two specific individuals, but we will be looking at two genes at the same time. So remember, you've got thousands and thousands of genes. I believe somewhere around 35,000. I could be way off. That just came to my head. And so we are looking at kind of how variation can be increased, even when we're just looking at two genes with two alleles. So again, we're not even looking at these genes having multiple alleles just yet. We're looking at two genes, two alleles, so 4 alleles in total, and all of the different ways these can be inherited. Okay, so there's a picture there. You've got one pair of chromosomes, so let's imagine that's chromosome pair 1, and it's got one specific gene on it, so a piece of DNA at a particular loci. In this case, all of the genes look like they're holding the same alleles, but that's not necessarily true. Okay, and then you've got your second pair of chromosomes, so that's down here, pair number 2, we'll just call that chromosome pair 2. And here they've shown us 3 genes. Now remember, the entire of that grey material is made of genes. It's just been very simplified to show us 4 bands. So we've got gene 2, gene 3, and gene 4. Alright, the 2 new words you're going to learn on this slide are linked and unlinked. So it's already set out for you here, but just to explain, if we're talking about gene 2 and gene 3 relative to each other, what we would notice is that any gamete that inherits this chromosome is going to inherit whatever allele is being held here, and whatever allele is being held here, and whatever allele is being held here. And the same for any gametes that receive this chromosome. Remember, 50% of the gametes will get this one, and 50% will get this one. And so those alleles will be inherited together every time. Obviously, we will talk about the very, very low chance that they may be separated by crossing over, but other than that, once in a blue moon kind of situation, these genes are what we call linked, and they will be inherited together. Whereas if we look at gene 1 and gene 2, we could get a gamete receiving this right-hand gene, this right-hand chromosome carrying whatever allele is here, plus this left-hand chromosome carrying whatever allele is here. We could get this right-hand chromosome being inherited with this one here. We could get the left one being inherited with this one or this one. So with our independent assortment during meiosis, we can get different combinations of chromosomes holding different alleles. So we would call these genes unlinked. So gene 1 and gene 2, for example, or gene 1 and gene 3, or gene 1 and gene 4. They are considered unlinked from each other because they can be inherited in different combinations because they are on different chromosomes. So pause here, process that information and make a note, please, and then we will move on. Okay, now we are going to start putting this idea into context, and what we're going to use is the very first example attributed to the man that discovered this idea. His name was Gregor Mendel. He was a monk in Austria, and he did his studies on peas. So he was looking at 2 different traits in peas. He was looking at surface texture, and he considered them wrinkly and round, and he was looking at colour, which he considered yellow and green. So, with respect to 2 genes, these 2 different traits, that each have 2 alleles for 2 different phenotypes that we can notice here, there are actually 4 possible pea combinations. You can have round and yellow, round and green, wrinkly and yellow, wrinkly and green. Oh, there you go. I read my own mind. Alright, when Mendel crossed yellow peas with green peas, he got these results. Okay, so this is the parent generation. One parent was green, one was yellow. And the filial 1 generation, F-I-L-I-A-L, means offspring or sons and daughters. So we call this generation F1. They were all yellow. But when he took 2 of the offspring of that generation and crossed them together, their offspring, F2, not only contained yellow offspring, but also green. We should be able to use that information to determine whether yellow or green is dominant. And no, you cannot use the idea that there are more yellow peas here to explain that. So pause and have a think about it, and I'll tell you the answer. Well, we can see here that this generation of yellow peas was able to produce an offspring that is green. And the only way to produce an offspring with a phenotype that is different to yours and not consider the idea of just random mutation as a cause is to say that that allele coding for that trait was hidden or masked in both of these parents. And for them to express one phenotype and mask and carry the other, that tells us that that carried allele is recessive. So each of them will be heterozygous for yellow and green. However, the green allele is being masked but a 1 in 4 chance of the green allele from one parent and the green allele from the other fertilizing to produce an offspring gives us 1 out of 4 offspring that are green. Okay, so that's following our expected ratio. Okay. Oh, sorry. Okay, so both of our F1 parents are heterozygous for colour. So they both have a yellow allele that provides them with their phenotype, but they both carry a green allele, which we know because they pass that on to their offspring. So in each of those parent cells prior to meiosis, DNA will double, and we'll have a homologous pair of chromosomes that looks like this. So if we're looking at the chromosomes that carry the colour gene, so that'll be these little black lines here, just this tiny gene on this chromosome. We've got 2 chromatids stuck together carrying that yellow allele from one of the original parents. Okay, and the other one carrying the exact same allele, because remember, these 2 chromatids are identical copies of each other. And then you've got the other homologous chromosome carrying the green allele, which is coded for by the lowercase y. And the other chromatid, identical, carrying another green allele. Okay, so that is one pair of chromosomes. This is one chromosome made of 2 chromatids, and this is another. All right, so after meiosis, we actually have 2 separations of genetic material, 2 divisions, and we end up with 50% of the gametes inheriting the chromosome that carries the Y allele, so the yellow allele, and we'll have 50% of gametes carrying the green allele. So remember, these 2 will separate into 4 different gametes. 2 of those will have these, and 2 of those will have these, and that'll occur multiple times over to produce hundreds of thousands of gametes. Roughly, well, not roughly, but exactly 50% of each. Okay, so recognizing that, oh goodness, that each of these parents, these yellow ones here that we know are heterozygous, produce 50% of their gametes with green, 50% with yellow. They end up with 1 offspring that inherits 2 greens, 2 offspring that inherit 1 of each, so they are heterozygous as well, and then 1 offspring that will be homozygous dominant. This is expected ratio, I'm saying. Okay, so again, we're just recapping what we know. Alright, so that's exactly what we just discussed. The 2 parents in the middle of that first diagram, the 2 heterozygous parents, will produce 3 yellow offspring, 2 that are heterozygous, 1 that is homozygous dominant, and 1 green offspring. This is the expected outcome. Alright, but now let's look at trait number 2, so round versus wrinkled. Well, he got a very similar result. So he crossed a round with a wrinkled, and he got a whole F1 generation of round peas. When he crossed 2 of those plants, 2 of those round plants, he ended up with round and wrinkled offspring. So just like before, we can see here that in order for this individual to look different to its parents, it inherited alleles that must have been masked in their phenotype. So they're carrying that allele, but it's not being expressed, which makes wrinkled recessive to round. And that's how we need to explain it. Not there are more round peas in the picture, but these 2 parents in the F1 generation must have been carrying an allele that they passed down to this child here that they did not express in their own phenotype, meaning that round was dominant. Okay, so just like before, both of those heterozygous parents have 1 chromosome with the round allele and 1 chromosome with the wrinkled allele. When that material doubles during interphase, they end up with 2 chromatids of each, and that will divide to form 2 gametes carrying round, 2 gametes carrying wrinkled. So, crucial to understand here that by the end of this stage, we have 2 heterozygous parents with 50% gametes that carry round, 50% that carry wrinkled, 50% are green, 50% are yellow. Okay, so just as we discussed, very similar outcome, a 1 to 2 to 1 heterozygous cross ratio. Okay, so that was a detailed introduction. I will now move on to the dihybrid cross.